Well, good afternoon, everyone, and welcome to this presentation on the use of functional ultrasound imaging for real-time whole brain functional mapping. And before starting and diving into the presentation, I want to thank SciTech Korea for giving me the opportunity to be here today and do this presentation to the Korean researchers community. So let me just very briefly introduce Iconeus, the company, before moving to the scientific part of the presentation. And Iconeus is a biotech manufacturing company. We're based in Paris, and we are a passionate team of neuroscientists, physicists, and engineers working hard to deliver the best possible product to our dear community of researchers. And we are very proud of our clients that are now spanning the globe from the Americas and Europe, but also in Asia, where we also have the precious aid of distributors, as you can see from the flags, including, of course, in Korea, where SciTech helped us bringing functional ultrasound to 2 clients already. But enough of the company, let's talk about functional ultrasound. So today's presentation will be structured in 3 main blocks. First, I'll talk about the basic principles behind functional ultrasound imaging. Then I'll give you an overview of the major applications of functional ultrasound through some examples of publications. And I will close the presentation talking about briefly the Iconeus One System and especially its software suite. So let's start from the basic principles of functional ultrasound imaging. And to understand the interest of using functional ultrasound imaging for the study of the brain, I always think that it's useful to briefly position it among the other imaging modalities that are available to study the brain and that are commonly used in neuroscience. Because, of course, you know, when you have to choose an imaging modality, you have to take into consideration its characteristics, for example, the ones that you see here on the left, like penetration, spatial resolution, temporal resolution, portability and versatility, and sensitivity. So, for example, if we consider fMRI and PET, we know they offer excellent penetration and coverage of the brain, but are limited in temporal resolution, portability, and experimental flexibility. And in particular, fMRI, which is a gold standard imaging technology for the brain, has relatively low sensitivity, making single trial detection and data processing difficult, while, for example, PET requires radioactive tracers and dedicated facility, so it's pretty hard to use and integrate in a daily experimental routine. On the other hand, we have optical imaging techniques that provide outstanding spatiotemporal resolution and good sensitivity, but their penetration depth is limited to the superficial cortical layers or to very small fields of view, again limiting their application for some specific experimental questions, right? And finally, we have the ultrasound-based family of technologies. For example, conventional Doppler ultrasound that you may already know has very good penetration, good portability, and spatial resolution, but it's limited in temporal resolution and sensitivity, again causing its usage to be limited to only some application, only the main vessels, and not widespread use in neuroscience. And that is why functional ultrasound, ultra-fast functional ultrasound, was developed to overcome these limitations for more widespread use. So functional ultrasound and Doppler ultrasound are based on the same physical principles of ultrasound Doppler. And so to understand why there is this difference between the two, we must start from how the technology works. So we have to start by saying that the brain metabolic demand, as you know, is met by the vascular system, where blood vessels supply neurons with nutrients to sustain their activity. So as you can see in the video, when neurons fire, the blood vessels vasodilate to allow more blood and nutrients to flow. This process is called neurovascular coupling and results in a local increase in cerebral blood volume when neurons are firing. And I'm saying this because the power Doppler signal that we measure when we send an ultrasound wave into the brain, as you can see from this blue wave here, what we measure with functional ultrasound is directly proportional to the cerebral blood volume, or CBV, which is the amount of red blood cells moving through the pixel at each specific time, as you can see here. So more red blood cells, more volume, more signal, as you can see from this depiction. But then if both conventional Doppler and functional Doppler ultrasound are based on this principle, how can they be so different in terms of sensitivity and temporal resolution? As you will see in this video, both conventional and functional ultrasound have a probe transmitting the wave placed above the animal's head, as you see here, but in conventional Doppler imaging, as you see on the left, the tissue is scanned sequentially line by line, restricting its application to the main vessels, while in functional ultrasound you see that the waves are plane waves, are flat waves, and so at each time point we collect 2D images, and you can already see here a big difference between these two images. But even farther, because functional ultrasound scans the brain plane by plane at ultra-fast frame rates, as you see here, it allows the acquisition of thousands of images per second. And over a period of 200 milliseconds, conventional Doppler ultrasound collects only 15 samples against the 200 samples collected with functional ultrasound. And these longer signals allow us to use powerful processing pipelines and to obtain much lower noise and therefore a much better sensitivity. And this difference in sensitivity is evident if we try to image the cerebral blood volume changes linked to neuronal activity in the case of a whisker stimulation. So you see here whiskers of a rat are stimulated and we see activation in the barrel cortex where there is a CBV increase due to the neurons firing after the stimulation. Of course, talking about functional ultrasound, we also have to talk about its versatility. Indeed, as you can see at the bottom of this slide, it can be used across several different animal species, not only rodents like mice and rats, but also in different animals like non-human primates, birds, and bigger animals like pigs and sheep, but also a variety of experimental setups. Indeed, functional ultrasound can be used on anesthetized animals, of course, but also in awake head-feet setups and awake freely moving ones. And it can be performed through the skull in mice, so even the preparation is very simple. And throughout this presentation, I'll try to show examples of study, showcase different animal models and different setups. So now to conclude this part, if we go back to our comparative table from which we started, now you understand better why functional ultrasound is a very good combination of these different characteristics and offer an excellent compromise for the study of the brain. And if we look now at all these features and give some numbers, because at the end this is what we care about, we have a tissue penetration that can be down to 4 centimeters using 6 megahertz probe, an excellent spatial resolution of 100 micrometers at 15 megahertz, a very good temporal resolution between 2.5 and 100 hertz, because we saw before how fast we can be, which is something that we cannot do with conventional ultrasound. It's very portable, and can be used in a variety of setups from anesthetized to awake. And it gives a 100-time increase in sensitivity compared to conventional Doppler ultrasound, with the ability to detect hemodynamic changes within a single trial, as you could see before from the whiskers being stimulated in the rat in the video that I showed before. So what does all of this translates into? Well, for example, we can look at an epileptic seizures induced by a pharmacological agent with a spatiotemporal resolution and sensitivity that are really unprecedented. You see here neuronal depolarization spreading through the cortex and in subcortical nuclei. Okay, so with this we close the first part, and let's now move to the major applications of functional ultrasound that we usually divide into three main blocks that are task-based functional imaging, functional connectivity, and stroke and vascular imaging. And I will start by task-based functional imaging, so the study of neuronal responses to specific stimulations. And I want to start with this simple study from 2017, which was a seminal study in the field, performed on an anesthetized rat that was presented with a visual stimulation. So here, a specific pattern of visual stimulation was delivered to the animal through the screen that you see placed in front of it, with a pattern of 30 seconds on and 30 seconds off for 5 cycles. And when we look at the neural activity of areas pertaining to the visual system, such as the primary visual cortex, the superior colliculus here in red, and the lateral geniculate nucleus in green, you see that their CBV changes, so cerebral blood volume changes in time due to the neural activity, closely mirror the pattern of the visual stimulation that here is indicated by these black lines. So something that with functional MRI would have taken hours to acquire and process can be done in tens of minutes using functional ultrasound. But of course, this is not all, right? And we have mentioned that functional ultrasound has also been validated in awake animals, either head fixed or freely moving. And here you see a freely moving rat performing a maze running task. So this animal is going to each side of this corridor to get rewards, while both his hemodynamic response in the hippocampus as his electrical oscillations in the hippocampus are measured, respectively, with functional ultrasound and local field potential recording. So here we have functional ultrasound combined with electrophysiology to study the responses of the hippocampus in this spatial task. And as you can see from this movie, in the initial run, you see these are the initial runs, functional ultrasound and LFP show a sparse activation in the brain, especially at the level of the cortex. But going on with the task, you will now see the intermediate runs. This response starts to shift from the cortex to the dorsal hippocampus, with a confinement of the responses mainly at the level of the hippocampus in the late runs, as you can see here. And this, of course, happens coherently with the animals learning the task. So this publication proved functional ultrasound potential for awake imaging, including the possibility of combining it with other recording modalities like electrophysiology. And finally, for this section, we said not only rodents, and indeed functional ultrasound has also been used on non-human primates, in this example macaques, where the neural activity in these two macaque monkeys was recorded during an eye movement task, so a visual oculomotor task, as you see in the title. And here functional ultrasound was able to reveal activity in the supplementary eye field that you see here and in the anterior cingulate cortex that is here. So areas that are involved in eye control, the supplementary eye field, and decision making, the anterior cingulate cortex, that were involved, of course, in this task, where the monkey had to fix and decide which side to go. Importantly, functional ultrasound could detect strong and localized activity in the supplementary eye field, as you can see here, just a few seconds after the start of the trial, and they could also reveal a propagation of activity starting from the anterior cingulate cortex all the way up to the supplementary eye field, as you see here in the video, like a wave spreading through the tissue, showing the direction of the information flow during this complex task. This brings us to the second major area of application, that is functional connectivity, which is a topic that's gaining more and more attention in the neuroscience field due to the several proof of its importance in different disease conditions, ranging from psychiatric disorder like depression and schizophrenia, but also Alzheimer's disease and autism, both as a possible underlying cause and biomarker. So to make sure that we are on the same page about functional connectivity, just a few words on what it is. So functional connectivity, as you can read on the slide, refers to the statistical dependence of activity between different brain areas, pointing to the functional relationship within the brain. So this means that there are brain regions, as you can see here, whose activity is highly correlated, while other brain region whose activity is not correlated at all. And these drive the emergence of specific networks that are activated at specific times or during specific tasks. And this pioneering publication of 2014 in Nature Communication showed for the first time that functional ultrasound could be used for the study of functional connectivity because regions known to be functionally correlated and connected, like these two blue and red regions, had CBV responses and CBV oscillations that were also temporally correlated. The extent of this correlation can be mapped into correlation matrices that we call connectivity matrices because we want to use them to study connectivity, and they can underlie and highlight the emergence of specific networks. So let's look at some examples, and the first one that I want to focus on is this paper from 2022 where the authors study emotional memory. So here the researchers used a simple fear conditioning paradigm and imaged the mice before training, after recent recall, and again after remote recall, and they found strong cue-evoked increases in connectivity across the fear network, especially at the level of the amygdala, between amygdala, hippocampus, and prefrontal cortex, as you can see in A, B, and C. And interestingly, over time, these connectivity patterns were refined. So if during recent recall, the main network was between the amygdala and the hippocampus, at remote recall, the network refined with stronger connection between hippocampus and prefrontal cortex, suggesting that the network reorganizes as memory consolidates. And they were also able to show that these network changes were correlated with the animal's freezing behavior, directly linking the connectivity to the memory expression. So in this study, they show how functional ultrasound can capture large-scale dynamics of memory encoding and retrieval in vivo, making it a valuable tool for the study of neural circuits for learning and memory. But then we also said how functional connectivity has been shown to be disrupted across several neurological disorders, and in these publications the authors set out to study a mouse model of the Charcot-Marie Tooth disease, as you see on the title, that in humans is a peripheral neuropathy causing decreased nerve conduction, paralysis, and tremor. And in this mice model, compared to wild-type and healthy mice, they were able to show that there was an overall increase in connectivity, both during resting states and during tasks, and especially a stronger connectivity between somatosensory cortex and hippocampus that was completely abnormal compared to wild type. And finally, it wouldn't really make sense to study connectivity at the level of diseases if we couldn't study it also at the level of pharmacology and pharmacological intervention. So to see how different drugs can impact on the normal functional connectivity of the brain or on the altered functional connectivity in a disease. So in this recent preprint, the researchers used functional ultrasound imaging in awake mice, as you can see here, to study how clinically relevant opioids like morphine, fentanyl, and methadone affect brain connectivity. They found that opioids consistently induced a marked reorganization of brain networks with a disruption of the cortical-subcortical connectivity. So instead of having connections between cortex and subcortical structure, there was an increase in the intracortical and intrasubcortical connectivity, as you can see from these maps. And importantly, these changes were dose-dependent and correlated with the development of analgesia, so the most known effects of opioids and their clinical effects of opioids. And so with this publication, the authors showed how functional ultrasound allowed them to reveal a highly reproducible and pharmacologically specific signature of opioid action in the awake mouse brain, which is something that would have been extremely hard to capture with any other imaging modality. Now, all the examples that I just showed were performed in 2D plane using 2D linear arrays to send the wave to the brain, and we can move across different slices to acquire volumes of the brain. But these procedures tend to be slow and not only adapted for the study of large and sparse brain networks. Sometimes we want to acquire and construct bigger matrices like the one that you see, and to overcome this problem, we have developed a 4D multi-array probe, the ones that you can see here, for the simultaneous multi-slicing of the brain that allow us to cover the whole mouse brain in just 2.4 seconds. So absolutely in line with the study of functional connectivity across the whole mouse brain. And as you can see from this figure from a recent publication, the use of this probe allowed the authors of the publication to identify several different networks, both cortical and subcortical, at once across more than 200 brain slices. So it's a very powerful tool. So this brings us to the last broad area of application, that is stroke and vascular imaging. So I'm going to show you these examples in which researchers used functional ultrasound to monitor cerebral perfusion in a mouse model of stroke, both before and after treatment. The results here highlight functional ultrasound's unique ability to provide real-time imaging of brain perfusion immediately after the stroke onset. You see here at T0, you can already see a dark area related to the ischemic lesion, as well as after intervention. For example, here you see 2 hours after the intervention. Notably, this study demonstrated that functional ultrasound can detect hypoperfused brain regions as early as 2 hours post-stroke, and the final lesion volume observed 2 hours after the stroke with functional ultrasound was closely correlated with the lesion volume observed with MRI 24 hours later. So these examples suggest how functional ultrasound could serve as an early predictor of tissue fate, enabling timely therapeutic decisions and also long-term monitoring. Now, of course, the possibility of imaging and studying the vasculature of the brain is extremely important to increase our knowledge of brain physiology and also to better understand several disruptive neurological conditions related to the vasculature. However, as you can see clearly from this drawing here, not all the vasculature is the same within the brain, and especially the structure and property of the vessels changed dramatically between the bigger and the smaller vessels down to capillaries. And actually, it is precise at the level of these smaller vessels, so small arterioles and venules, and at the level of capillaries, that most vascular disorders show their stronger effects. So it's never at the level of the main arteries and main veins that we see the disruption in activity, or like this happens only when the disease is very advanced. And with functional ultrasound, we mentioned before, we can image down to 100 microns of resolution, which allow us to look at arterioles and venules, the bigger ones, but this may not be enough, right? If we want to really study the small vessels that could be the cause of some diseases. But luckily for us, we have the possibility of using a contrast agent with functional ultrasound to increase our spatial resolution down to 10 micrometers for the study of capillaries, and to go from images that are like this, so this is what I've shown you so far, to images that are like this. So with a spatial resolution of down to 10 micrometers, allowing us to study even the smaller vessels. This technology is called ultrasound localization microscopy, or ULM, and let's see how it works. Now, ULM relies on one key ingredient that are microbubbles as contrast agents. Okay? These gas-filled bubbles are injected into the bloodstream, and when they are exposed to ultrasound waves, they produce a strong echo that stands out against the rest of the tissue. And because we acquire thousands of images over time at ultra-fast frame rates, we mentioned it at the beginning, we can pinpoint the exact location of these bubbles with precision beyond the diffraction limits. And by tracking these bubbles frame by frame, we reconstruct a super resolved map of the vasculature, revealing even the smallest vessels, and we can look really at something like this. So these images, I think, are really breathtaking. So I'll shut up to let you look at it. And actually, what's really important and cool about this technology is that we can not only look at the morphology of the vasculature, but we can also measure the blood speed and the direction of the blood. Informations that can be used across different studies. So here you see an example of ULM for the study of Alzheimer's disease. So here the authors investigated early microvascular changes associated with Alzheimer's in 5x FED mice model. And they were able to assess that at 3 months, the transgenic mice exhibited a significant reduction in the blood flow velocity within hippocampus and entorhinal cortex compared to controls. And this was sustained at 6 months, but even more, those same regions displayed a worsening of the condition with decreased vascular density and increased tortuosity of the vascular network. So you see, for example, how ULM was used for the study of a neurodegenerative condition. Another example is this recent publication in which researchers imaged the trigeminal ganglion, which is a small and deeply located structure in this case of a rat at 12 millimeters of depth. And because of this, prior to this study, the trigeminal ganglion microvasculature was only described ex vivo. While here the authors, in the context of neuropathic pain, used ULM to reveal the trigeminal ganglion intricate vascular features, including a very dense vascular network, high blood speed, and multiple direction of flow. Now, again, these examples are all examples performed on a single 2D slice. But what could be a game changer would be to be able to image an entire volume using ultrasound localization microscopy. So having this great resolution to dissect the entire and intricate three-dimensional network of the vasculature. And this is why we developed a specific probe to do this that's called 4D RCA probe that allow you to observe the entire mouse brain at unprecedented 10 micrometers resolution. And from this, you can even do as far as doing single vessels analysis in the entire brain. And with this, we have arrived to the last part of this presentation. And I know while this presentation is supposed to be focused on the science behind the technology, we cannot really avoid to talk about the Iconeus One platform. Because the Iconeus One platform is not just a piece of equipment, but it's really an integrated and dedicated system for the study of the brain in the neuroscience field. So let's see some characteristics of this platform. First of all, as we mentioned at the beginning, it's highly mobile and versatile, requiring minimum lab space, and can be used for several and shared research projects across facilities and animal models. Of course, we give the possibility of synchronized functional ultrasound acquisitions with other modalities, so there are BN support for synchronizations with electrophysiology, optogenetics, EEG, LFP recordings, whatever you need. But most importantly, and what I want to focus a bit more on, is the full software suite that's integrated in the system to really fast forward your analysis. The software suite is composed of 3 different software, the icons you see here, that are IcoScan for scanning purposes, and so for acquisition. And of course, it allows you to acquire 2D and 3D recording in absolutely a plug and scan way. So it's really easy to use and to learn, but at the same time, highly customizable according to your needs. Then, most importantly, we have IcoStudio, which is our visualization and exploration software. In IcoStudio, you have the possibility to automatically register your vascular scan to the Mouse Allen Brain Atlas or to the Sigma Rat Atlas, and to use the regions of interest directly within the Atlas space, increasing reproducibility and robustness. You can also compute connectivity matrices as a seed-based map, so everything that we mentioned before can be done directly within the software, exploring a number of parameters in a simple way and in a few clicks to see which are the best for your specific data. You can do something similar to reconstruct activation map after specific stimulations. Here you see again a whisker stimulation, and you can extract the signals and do further processing. And of course, you can visualize your super resolved vascular maps, as you can see here. However, this wouldn't be enough without the presence of our last software, IcoLab, which allows you to apply all the explored parameters of IcoStudio to all of your scans. So here, IcoLab, you load all of your scans and batch process everything to obtain results and publication ready and group analysis. You can also do advanced motion correction, reconstruct the super resolution maps, and soon we also plan to include advanced statistical analysis for complete group comparison and really publication-ready results. So let's conclude. This is my last slide, and I want to leave you with some take-home messages. So we said that we would talk about the basic principles behind functional ultrasound, and we have seen that thanks to the plane wave technology, functional ultrasound imaging grants high spatiotemporal resolution and sensitivity for the study of large-scale brain activity and vascular structure. We also said that we would look at the major applications of functional ultrasound across different examples from the literature. And indeed, you saw how functional ultrasound can be applied across animal models and experimental settings, from studies that are the most variable, from basic physiology to complex cognitive processes to brain disorders and even pharmacological studies. And I said that I would conclude by talking about the Iconeus One system and its software suite. And indeed, I showed you how the Iconeus One system and the software suite form a really integrated and complete system, allowing researchers to customize their setup and move quickly from their experimental ideas all the way through results. And with this, I wanna thank you for your attention and thank you again to SciTech Korea for the opportunity.
안녕하세요. 반갑습니다. 저는 어, 이 아이코니우스 원 시스템의 한국 본국을 맡고 있는 주식회사 사이텍 코리아의 학술이사 이정윤이라고 합니다. 지금 이어지는 Q&A 세션으로 넘어가기 전에 그 아이코니우스 원 시스템에 대해서 간략하게 소개해드리고 다음 Q&A 세션으로 넘어가도록 하겠습니다. 지금 소개해 주신 내용 중에 있는 아이코니우스 원 시스템은 세 가지 컨피규레이션이 있고요. 그 가장 하위 성능을 가진 아이코니우스 원 익스플로러부터 그 상위 버전인 익스밴드 그리고 엑셀 버전이 있고요. 그 3D, 4D 프로브를 사용할 수 있는 엑셀 그 아이코니우스 컨피규레이션 시스템 엑셀 버전만 가능하고 각각은 모두 필요에 따라 업그레이드가 가능한 시스템들입니다. 또한 그 각 시스템에 맞춰서 그리고 실험 목적에 따라서 그 실험 동물의 종류에 따라서 적절하게 사용할 수 있는 여러 가지 프로브 또한 아이코니스사에서 제공하고 있습니다. 그리고 아이코프라임 4D 멀티 어레이와 RCA 프로브는 선택하셔서 별도로 또 사용하실 수 있습니다. 이 부분에 관련돼서 기술적으로나 궁금하신 점이 있으시면 사이트 코리아로 문의 주시면 답변 드릴 수 있도록 하겠습니다. 그리고 금일 세라 로만지 박사님께서 발표해 주신 발표 내용은 전체 공유는 드리지 않고요. 관심이 있으신 분은 사이트 코리아로 이메일 연락 주시면 저희가 공유 드리도록 하겠습니다.